Hello everyone, my name is Collin Hill and today we'll be discussing utilizing mutagenic compounds as broad spectrum antivirals. So first we see that all RNA viruses besides retroviruses utilize the RNA-dependent RNA polymerase for both viral genome replication and for the synthesis of mRNAs, which will be later translated into viral proteins. The RNA-dependent RNA polymerase has a notoriously low fidelity, and this leads to the high error rate of RNA viruses, which can cause for a high adaptive ability of RNA viruses to a changing environment, which can make it difficult to develop antiviral drugs and to develop effective vaccines. Also, we see that RNA-dependent RNA polymerase lacks an analogous cellular process in eukaryotic cells. So what that means is in eukaryotes like us, we lack the ability to convert RNA into RNA. And because of this fact, or these facts, it makes RNA-dependent RNA polymerase a prime target for developing antiviral drugs. So one mechanism of developing antiviral drug to target RNA-dependent RNA polymerase is through lethal mutagenesis. So because of the high mutation rate of RNA viruses, it causes the viral population to live near its error threshold. And at a mutation rate at above this error threshold, it'll lead to the rapid accumulation of deleterious mutations, which is known as error catastrophe. And this causes a significant decrease in the mean fitness of a viral population, which can lead to a decrease in infectivity. And because of this, what we can do is we can take mutagenic compounds and we can use them to push a virus over its own error threshold and induce an error catastrophe. And this could potentially allow us to use mutagenic compounds as therapeutic drugs to treat viral RNA infectious diseases. So in this study, we examined 3 different nucleoside analogs: ribavirin, favipiravir, and N4-beta hydroxycytidine, otherwise known as NHC. Each of these drugs have been previously shown to be mutagenic to different viruses and have been shown to exhibit antiviral effects against several different viruses. However, we'll be examining some new viruses. And what we did with each of these different compounds is we created a drug panel with a variety of different dosage concentrations, and we examined their effects on 3 different viruses in cell culture. So that was MERS coronavirus, Zika virus, and the Cross virus, each of which are from a different genus. And we examined their not only their antiviral effects, but also their mutagenic effects of the compounds on these 3 different viruses. In addition, we also tested this drug panel on 8E5 cells, which host a defective proviral HIV genome that is expressed but not able to replicate. And then what we did is we measured the cytotoxic effects of the compounds on the 8E5 cells, but we're also able to sequence the expressed HIV RNA in order to measure the transcriptional and DNA mutation rates that could potentially be caused by the compounds. In addition, we also tested an additional compound known as EIDD2801, which is just an NHC prodrug. And the only difference between NHC and this prodrug is the addition of an isopropyl ester group, which causes this prodrug to be orally bioavailable, which allowed us to do an in vivo mouse experiment, which was a MERS coronavirus mouse model. And what we did is we gave EIDD2801 orally at a concentration of 500 milligrams per kilogram daily to these mice. However, their dosage regimen were started at different time points, whether it be prophylactically before MERS coronavirus infection or at various different time points after MERS coronavirus infection of the mice. So then for each of the different cell culture experiments looking at the 3 different viruses, we measured infectivity either by using a plaque forming assay or a focus forming assay. And then for the 8E5 cells, we measured cytotoxicity using automated cell counting. Then for all of the different experiments, we did viral RNA extraction, and for the mouse model experiment, we extracted mice mRNA at the same time as we extracted the viral MERS coronavirus RNA. And then using primers that are specific to each virus or that were specific to the upregulated genes of the mice, we were able to synthesize cDNA from the extracted RNA. And these primers also had degenerate block of 11 nucleotides that were to serve as the primer D for future analysis. So then after PCR implication of the cDNA, we did Illumina MiSeq next generation sequencing. And then after those results, we did template consensus sequence analysis of the sequencing results, and we're able to create a template consensus sequence, which is able to greatly reduce the method error rate of deep sequencing while maintaining a high deep characterization of the viral population. And this was this was done in order to accurately determine the mutation rates, as well as to validate the detection of rare mutants. So what we found with NHC in each of the 3 different cell culture experiments with the 3 different viruses, as we can see on the left-hand side, we see the total error rate, and on the right-hand side, we see the loss of infectivity. We saw that NHC exhibited both antiviral and mutagenic effects on each of the 3 different viruses. And then we're also able to correlate that this loss of infectivity or antiviral effects is from this increase in error rate. So we're able to predict that NHC acts through a mechanism of lethal mutagenesis. And then looking at the other 2 compounds, ribavirin and favipiravir, in the cell culture experiments, neither one of them exhibited either antiviral or mutagenic effects in MERS coronavirus or Zika virus. However, for all 3 of the different compounds, we did see an increase in the total error rate and antiviral effects in lacrosse virus. However, ribavirin and favipiravir to a much lesser extent than NHC. So then, looking at the mutagenic profile of NHC on lacrosse virus, we see that NHC primarily increases the CTU and G to A transition rates. And to a lesser extent, we see an increase in the A to G and U to C transition rates as well. And then for ribavirin and favipiravir in lacrosse virus, we see a more modest increase in the C to U and G to A transition rates as well for both of these compounds. And then what we're able to extrapolate from this mutagenic profile of NHC specifically is that we're able to verify that NHC acts primarily as a cytidine analog and a uridine analog. And what we can see is depending on what strand NHC is incorporated in, that's how we see the different types of mutations from sequencing. And because NHC primarily binds to guanosine, we mainly see the largest increase in the G to A and C to U transition rates. So looking at the C to U transition rate in MERS coronavirus, we see that NHC increases its C to U transition rate in a dose-dependent manner. However, with ribavirin and favipiravir, we do not observe any increase in the C to U transition rate. And then we see similar results in Zika virus. And then, however, with lacrosse virus, at a dosage concentration above 10 micromolar, we see a plateau in the increase of the C to U transition rate for lacrosse or for NHC. And then we also see a modest increase in the C to U transition rates for both ribavirin and favipiravir, again in lacrosse virus. So looking at the 85 cell culture, we're able to look at the cytotoxic effects of each of these three different compounds just by measuring the cell count over a long period of time when they're cultured with these drugs. And what we see is that NHC in blue has an intermediate cytotoxic effects to ribavirin in orange and favipiravir in green. However, something interesting that we found was that NHC drastically increased the air rate of the HIV that was produced within these cells. So what this could be indicative of is an increase potentially of the DNA mutations, which could cause NHC to be potentially carcinogenic. But it could also indicate that NHC is causing an increase in the misincorporation of RNA polymerase II, which could be increasing the transcriptional error rates of these cells. And that's exactly what we observed, the latter one, which is because we only saw an increase in the T to C and C to T transition rates, which occur, both of those only occur on the positive sense DNA. It provides evidence that we are really only seeing an increase in the transcription error rate of caused by NHC, and less evidence for NHC causing DNA mutations. And then some further evidence we saw that NHC increases only really the transcriptional error rate in 85 cells is that over time, we see that the error rate caused by NHC plateaus after day 6. And if NHC was primarily causing DNA mutations, we would see a gradual increase over time because there's no selective pressure on this proviral HIV. So we would only be seeing an increase over time, but this plateau is evidence that we're really only seeing transcriptional error rates. And by looking at this increase of transcriptional error rates caused by NHC, it provides some evidence that the cytotoxicity that we're observing from NHC could be caused by this mutated mRNA, which could be causing downstream truncated proteins and misfolded proteins that could otherwise cause cytotoxic effects, but further experiments are needed. And then, so now we're looking at the mouse model of MERS coronavirus. And again, this is using an NHC prodrug, EIDD2801. And the main thing we wanted to show here was that even in vivo, we're able to see both the antiviral effects of NHC on MERS coronavirus, but we're also still able to see significant mutagenic effects, still indicating that NHC's mechanism of action in vivo is through lethal mutagenesis. However, also importantly, we simultaneously sequenced upregulated mice genes. And what we observed was that there was not a significant increase in the errors of the mice mRNA. And this kind of goes contrary to the 85 experiments that we saw, because we don't really observe the transcriptional increase in the transcriptional error rate or DNA error rate in vivo. But again, this could be because there's differences in the dosage concentrations in vivo versus cell culture. So in conclusion, we're able to observe that NHC acts as a broad spectrum antiviral through a mechanism of lethal mutagenesis. And we're able to see that ribavirin and favipiravir can act as an antiviral through lethal mutagenesis, but really only for certain viruses, and in our case, it was Lacrosse virus. However, there could be limitations with these assertions because the cells that we used could have potentially caused cell-mediated drug resistance. So further studies repeating the experiments with the different viruses would be needed using different cell lines. And then also we observed that NHC does not exhibit significant short-term toxicity in vivo in the mice model, increasing the transcriptional error rates. But over long-term in cell culture, in the 85 cell culture, we did observe an increase in primarily the transcriptional error rate. And there is still potential for NHC to be causing DNA mutations However, in order to really prove a negative result, more experimentation is needed, like doing DNA sequencing and other experiments. So I would like to thank Swanstrom Lab, especially Shuntai, my lab mentor, as well as Sabrina and Matt for helping out with some of my library preps, as well as Barrick Lab for helping out with the MERS coronavirus experiments, both the cell culture and the mouse model. And then I'd like to thank Lazier Lab for helping out with the Zika virus cell culture experiment, and the Rocky Mountain Laboratories from NIH for helping out with the lacrosse virus cell culture experiments. And then I'd also like to thank George Painter from EIDD for supplying EIDD 2801, as well as Ray Shinazi from the NIH AIDS reagent program for providing NHC for our experiments. So I'd like to thank you for listening to my talk, and that completes my thesis.